Good afternoon, and welcome to the Brain and Behavior Research Foundation's Meet the Scientist monthly webinar series. I'm Dr. Jeff Borenstein, and I have the honor of serving as the President and CEO of BBRF. Today, Dr. Galen Missig will present, Does early life inflammation contribute to the risk for developing psychiatric conditions? BBRF is the world's largest private funder of mental health research grants, supporting transformative discoveries in order to develop improved treatments, cures, and methods of prevention. The high quality of the research we fund is made possible by the BBRF Scientific Council. This group of 180 prominent mental health researchers reviews each grant application and selects the most promising ideas with the greatest potential to lead to breakthroughs. The Scientific Council guides the Foundation to fund creative and impactful basic, translational, and clinical research relevant to the whole spectrum of mental health. One reason that research funded by BBRF has had such a great impact is because we do not limit our focus to one illness or condition. Since 1987, the Foundation has awarded more than $430 million to fund more than 6,200 research grants around the world and across a broad spectrum of brain illnesses, including addiction, ADHD, anxiety, autism, bipolar disorder, borderline personality disorder, depression, eating disorders, OCD, post-traumatic stress, schizophrenia, as well as suicide prevention. Today, I'm delighted to introduce Dr. Galen Missig. Dr. Missig is Senior Scientist in vivo pharmacology at Cerevel Therapeutics. He was a 2018 Young Investigator. Our webinar will begin with Dr. Missig's presentation, which will then be followed by a Q&A. To submit your questions, please use the Questions tab on the control panel on your screen. Feel free to submit your questions at any time. Following the presentation, I'll ask as many questions as possible in the time allotted. And now, I'm pleased to introduce Dr. Missig. Galen, the floor is yours.
Wonderful. Thank you so much. Thank you so much for that introduction. And, it really is, I really feel lucky to be here. And I feel, really touched that so many of you have shown up. And, I think this is both, an incredibly interesting and important topic, but I'll admit I'm rather biased. And so we're going to be talking about whether early life inflammation contributes to the risk of developing psychiatric conditions. Now, before I begin, some important disclosures and some important information about myself. So last December, I got a great opportunity to work for a company called Cerevel Therapeutics. It's a new company developing drugs for both neurologic and psychiatric conditions. But the work I'm going to tell you about here today comes from before that time when I was a researcher at McLean Hospital in the laboratory of Dr. Bill Carlazon. And the other thing to know really about this field of how really inflammation or the immune system affects the brain, it's still very much an emerging field. And we often have a lot more questions than we do have answers. And what I'm going to share with you today is really some of my work that puts together kind of a few of these pieces and how they might fit. So what we were particularly interested in was autism spectrum disorder, or ASD. Now, the most recent estimate is that 1 in 44 children born today will go on to be diagnosed with ASD. And this is even greater if that child is a male, because there's almost a 4-to-1 male to female predominance. Now, it's important to know that autism has really been defined by the presence of a set of behavioral features. These include things like social impairments, communication deficits, and the presence of what are known as repetitive or stereotyped behaviors. And now the exact kind of presence of what particular behavioral features has changed over the years to improve diagnoses. But classically, it's been made through the presence of behavioral abnormalities. But what there is a widespread appreciation for is that many of the co-occurring medical features, these include things like sensory abnormalities, sleep dysregulation, epilepsy or seizure disorders, and things like gastrointestinal dysfunction. These may actually be stemming from some of the same underlying biology. And that to really understand and see the bigger picture, we need to encompass these co-occurring features as well when we're talking about this disorder. Now, we were particularly interested in a potential immune subtype of autism. And what I mean by this is some form of autism that's either characterized by the presence of some form of immune abnormality or some potential even contributory role of immune dysregulation to this disease. Now, the evidence for this comes from several different directions. Quite simply, if you look in the blood of those that have been diagnosed with autism, you'll find that they'll often exhibit either increased or decreased levels of various inflammatory markers known as cytokines. And these are, generally increased or decreased, and it's hard to get an exact clear picture of what's going on, but we often see changes in the peripheral immune system. Now, if we go on and look up into the brain, we can see some signs for immune dysregulation within the brain as well. So within the brain, there is a resident primary immune cell known as microglia. And what studies have observed is that in individuals that have had autism, these are post-mortem analyses, they can see that either these microglia are changed in shape or changed in number. And this is generally what happens in response to ongoing inflammatory processes within the brain. And another line of evidence comes from a number of genetic studies. So there's been an intense interest in the genetics of autism, especially in recent years. And in studies that have looked at, looked genome-wide and looked at what genes in particular are changed in what we call their expression, or how much of that gene is present in the brain. One of the overall pictures that's come to light is that there are two major clusters of genes that seem to be changed. There's the top cluster here, which is related to more of communication by neurons or synapse. And then the bottom cluster, which is more related to immune processes. But it's important now to really kind of take a step back and ask, well, all right, well, does this make sense? Should we expect to find some form of inflammation in a condition like this? Is there any kind of plausible explanation? And I think there might be, because of really a number of findings we've made in really the past couple decades, suggesting that the immune and the nervous system are much more interconnected than we once thought. So in fact, we often find that these two systems are essentially speaking in the same language. So what I mean by that is, for things that we used to think that were primarily immune molecules, they keep turning up in the brain and play an important function. So for example, those cytokines that play an important role in inflammatory responses, well, they actually play an important role in stimulating the generation of new neurons in a process known as neurogenesis. Chemokines, which are these molecules important for actually guiding immune cells to the site of infection. Well, they're actually essential in the embryonic brain, guiding a set of neurons to the correct final location. And finally, we've seen how things like the complement system. So complement is used by the immune system to tag invading microbes, viruses, bacteria, telling the immune system that these need to be eliminated. Well, the same process is happening in the brain, where complement is actually tagging connections between neurons known as synapses, telling them that they need to be eliminated. And the reason why I'm telling you all this, and the reason I'm emphasizing this interconnectedness between the immune and the nervous system, is because now you can imagine a situation where during some key period of development, such as the growing embryonic brain, where millions upon millions of neurons have to connect in the right location at the right time. It's an incredibly complex process. And if you throw something in like immune activation, you might be able to alter that trajectory of the developing brain to result in something like a neurodevelopmental disorder. And in fact, we might actually see this manifest in an epidemiologic association between various types of inflammation during pregnancy and an increased risk that child of that pregnancy will go on to be diagnosed with autism. And now this is not only true for both bacterial and viral infection, but also things like autoimmune disorders as well. Now, it's important to note, and I just want to put an important caveat right here, is that generally, this is generally an increase in the risk. So, studies vary from odds or hazard ratios of 1.5 to 2. So you could say it's, at most, it's essentially doubling the risk. So, even though there's a presence of a bacterial and viral infection during a pregnancy, most of the offspring are, unaffected and will end up neurotypical. And a second important caveat to this is that this is not only true for autism. There's been similar lines of evidence for really a wide variety of psychiatric conditions, including ADHD, bipolar affective disorder, and especially schizophrenia as well. Now, in an attempt to model this in the laboratory, the late Paul Patterson had developed what he had termed a maternal immune activation model. So to look at this in really just a highly controlled way, you are able to time mate mice, and so you know exactly when they get pregnant, and then you can use something to mimic a early kind of viral response. It's called an innate immune response. In this case, they used poly-IC, and they injected it in the middle of pregnancy, and then looked at what happened to the offspring of that pregnancy. And what he found was that the mice of that pregnancy had abnormal social behaviors, behaviors related to communication, and abnormal repetitive behaviors as well. And we'll go in exactly how these particular behaviors are measured, but this was the essential model, and this was really a lot of the research that was coming out when I had joined the field. And our initial goal was to really kind of replicate and extend this model, but we ran into one significant problem. So don't worry about this too much, but this is social interaction behavior. And this is really one of the more hallmark behavioral features. And all I wanted to show you here is that we tested a lot of mice and we did not see an effect using this model. And what's really been amazing in just the past couple of years is that there's been a lot of research suggesting that things that you might think of as relatively subtle factors, this includes, the exact caging environment, or things like the exact composition of the gut microbiome of that pregnant mouse, can really have dramatic effects on whether or not you see this phenotype. But I had gone a slightly different direction. Going off of the idea that that initial immune insult may essentially sensitize the system to any kind of other later hits, we had developed a two-hit model. So we did this classic Paul Patterson model where we injected that viral mimetic poly-IC on embryonic day 12.5 of pregnancy, which is roughly the middle end of pregnancy for a mouse. And then, did a second immune insult on postnatal day 9. And this time we used LPS, which is more similar to kind of a bacterial response. It's actually the cell wall of E. coli. And it's important to note here that it's a little bit difficult to directly translate across humans to mice, because in this case, essentially the third trimester of essentially brain development, the rough equivalent occurs during the first 2 weeks following birth in a mouse. So this is roughly equivalent to really kind of the end of the third trimester in terms of when you're translating this back to humans. So we had these two different hits, the prenatal hit with poly-IC, postnatal hit with LPS, and this gives us this kind of 2x2 design that you can see in the top right panel, where we had, either vehicle, or that's essentially saline. We did the prenatal hit alone, the postnatal hit alone with LPS, or the prenatal plus postnatal hit. And then we assessed them on a battery of different behavioral tests. And what we found was that this time we could recreate a number of those altered behaviors. So on this is what's known as the social interaction test. And so you can see that there is that one mouse behind a cage. That's the control mouse that you allow it to it's a way to control for how well that mouse is interacting. And then you have your experimental mouse there on the bottom left there. And then you can quantify how much that mouse is interacting with your control mouse. And what we found was that for male mice that were treated with that postnatal LPS, they had a significant decrease in social interaction. And this was true for males, but not for females. On a test of anxiety-like behavior, so this is called the open field. So this is a very commonly done anxiety-like behavior test where it's essentially a large box, and mice will tend to actually hang out around the periphery. Being animals that are commonly preyed upon, they prefer the safety of the periphery, but they will also have a desire to explore the entirety of the arena. A mouse that hangs out mostly in the corners or around the periphery is said to exhibit an increase in what we term anxiety-like behavior. And what we found is in this model, as you can see in both red and purple, the mice that were treated with that postnatal LPS had enhanced anxiety-like behavior. And it was true in both males and females within our two-hit group. Now, communication-like behavior is admittedly something that's very difficult to study in a mouse. And you might think of as mice as normally quite quiet. Well, they actually do emit calls, and they actually do, they're just kind of well above our hearing. So mice will emit calls, and our hearing tops out around 20 kilohertz, but mice will emit calls somewhere between 40 to 100 kilohertz. And they'll often emit calls under certain particular scenarios. In this case, you can essentially, if you separate a pup, so a young mouse that's only 10, 12 to 16 days old, it will actually emit calls to try and get its mother to retrieve it to bring it back to its nest. You can then use a specialized microphone there on the left to record these calls. And so you can see here, this is a vehicle animal emitting calls to get its mom to retrieve it. But don't worry, they're only separated from their mom for 3 minutes. So, and you can see when we did the prenatal treatment, we didn't get much of an effect, but we did that postnatal treatment with LPS, and now you can see there's actually a significant increase in the number of calls that these pups are emitting. You can see a slightly stronger effect on postnatal day 16 in those mice that had received both of these hits. So this is when, this is a form of altered communication like behavior when they are, still very young. What about when they get a bit older? Well, to do that, we did a test that actually only works in the males because what we did is essentially you can take a Q-tip and you can take that's laced in female mouse urine, and that male mouse will spend a lot of time investigating that Q-tip, as you can see on the left, and then emit a number of calls in response. And so you can see the number of calls there on the left, and the time investigating that Q-tip on the right. And this is true unless you are one of those male mice that was treated with that postnatal LPS. Now you're no longer interested in that socially relevant cue, nor do you emit nearly as many calls in response. So less interest and less of a response to a socially relevant cue. So overall, we were able to recreate a number of those initial findings with alterations in a number of communication-like behaviors using those ultrasonic vocalizations, decrease in social behaviors, and an increase in anxiety-like behavior. And there was some sex effects where there were stronger effects in the male versus the female mice. But we are, very avid users of these behavioral approaches. But we're also very well aware they have a number of significant limitations. First, they often look very different from what you're going to measure in humans. You can't put a human in an open field and expect them to behave in the same way you would a mouse. And second is that they often lack kind of a ability to repeat the tests, and they have just such a very different And second, they often lack, they're based kind of fundamentally on kind of anthropomorphizing what's going on. So you just because you can't really ask a mouse how it's feeling or doing. So you have to kind of infer what it's doing in a particular scenario. So in a search to find measures that are more similar and more objective and what we're terming kind of more translational, we've started looking at some other metrics, including using a EEG EMG telemetry system. So EEG, or electroencephalography, is a way to look at and record kind of overall brain activity from populations of thousands of neurons. Electromyography is a equivalent, but measuring muscle activity. In this case, we measure from the trapezius, but we also are able to measure temperature and activity. And we can do this using a remarkable kind of set of technology. These are entirely kind of wireless probes that can be implanted in either a mouse or a rat, and they can then freely move around. Here you can see a mouse right next to one of these transmitters, and on the top left there. And on the top right, you can see that you can use that EEG and EMG information to divide it into what is essentially known as vigilance states, or sleep-wake states. So you can divide it to whether the animal is awake and slow wave sleep or REM sleep. We can also look at things like looking at overall activity patterns. So you can look at these, what are termed diurnal rhythms. And you can see in the bottom left there, you can see in that white phase, this is actually during when the lights are on. So mice are actually nocturnal. So they tend to sleep during the day and be awake at night. And so that's what you can see, is that kind of daily activity pattern. So what we did was a fairly straightforward experiment. Using that two-hit model that we had characterized with those behaviors, we then implanted mice when they were 6 weeks old. So this is, roughly kind of a juvenile mouse. And then we just simply recorded and looked for what was altered at baseline at postnatal week 7. And what we found was a shift in overall sleep architecture, where we saw a decrease in time spent awake and a selective increase in slow wave sleep with no overall change in REM sleep. So some form of alteration in that sleep architecture at baseline. But I think perhaps more strikingly, I noticed something that was really readily apparent when I was scoring that sleep on some of these EEGs. And here's a recording from one of these mice, or one. And as I hope you can appreciate, that really kind of regular, high amplitude EEG activity is actually what's known as a spike wave discharge. And this is actually a form of epileptiform activity. Here you can see that spike wave discharge on example 1 there on the top. That actually occurred while the animal was asleep. Example 2 there occurred while the animal was awake. And you can see on the EMG, there's actually a brief kind of flattening of the EMG, because what actually happens is during this spike wave discharge, that mouse actually just kind of pauses during when it occurs. And now we were actually very kind of excited to see this, because as you may know, autism and epilepsy share a lot in common. In fact, you can say roughly about 1/3 of those with childhood epilepsy are diagnosed with autism, and inversely, the same proportion remains when it's about roughly 1/3 of those with childhood autism have epilepsy. And just as interesting, if you look at those with autism that have no known diagnosis of a seizure disorder, and you do long-term and EEG recording, one or two studies have actually demonstrated that looking at long-term recordings of children with autism, you can often see epileptiform activity, even in the absence of a diagnosis of a seizure disorder. And that this epileptiform activity is actually more common during sleep. And to me, what I think this says is that there is just some kind of commonality between the changes that we see going on in the brain in epilepsy and the changes we see going on in autism. So if we look in our model and looked at where these spike wave discharges are coming from, you can see that it wasn't all of the mice that were exhibiting these spike wave discharges, but it was primarily coming from those that had received that postnatal LPS. It wasn't all of the postnatal LPS mice, but many, but it was fairly exclusive to those that had received it. Additionally, if you look at the graph on the right, this is looking at when those spike wave discharges occurred. And you'll see that it primarily occurred during that the light phase, and so that the light phase, which is Zeit hour 0 through 12 there, and this is actually when the animal was sleeping. So the last thing we looked at is that you can kind of get an overall sense of how an EEG is changed and how generally the characteristics of EEG by doing something called a power spectral analysis. So you can kind of think of this as just kind of another kind of phenotype of the brain. And what you can do is you can essentially break down that EEG wave, which is a composition of many waves, into its various component parts. You can look at whether it's, some of the relative power in kind of the higher frequency, low amplitude wavelengths of beta and gamma, all the way down to the high amplitude, low frequency wavelengths, delta, that predominate during sleep. So when we looked at that EEG power spectrum, and we looked at how it was changed, what we found is a distinctive shift. So if you focus on the purple bars, which is our two-hit group, this is how they changed from our control animals. And you can see that we have this from lower frequency on the left to higher frequency on the right. And what we saw was an increase in some of the lower frequencies like theta, and a decrease in the mid frequencies like alpha, and higher frequencies like beta and gamma. On the right, you can see this is just another way of showing this same shift in EEG spectral power. Now, when I try to make this kind of in context with humans with autism, the closest parallel is to look at a EEG resting state studies. And the results have been, incredibly variable. But in some regards, this is kind of an all-comers, where we know that autism's a, incredibly heterogeneous disorder, and it's likely due to multiple causes. But when they tried to see kind of the bird's eye view, what they saw was a distinctive shift in power, which is kind of this U-shaped change in power distribution. And now mapping on what we found is we saw about 2/3 of this, where we saw an increase in the lower frequencies, a decrease in the mid frequencies. But what we found was actually a decrease in some of these higher frequencies, which is intriguing because it says it could be a particular kind of signature of this particular immune activation or particularly maybe just in these mice. So in summary, what we found is that using this kind of Two-Hit model, we were able to see a number of altered metrics using these kind of neurophysiologic measures with increases in slow wave sleep, that really distinctive epileptiform activity, and that shift in EEG spectral power. Now, that was looking at the activity of, thousands to hundreds of thousands of neurons in aggregate. What about what happens to an individual neuron and an individual neuronal circuit? Can we see the effects of this model at the level of individual neurons? So in a collaboration with Vadim Bolshakov and Yan Li, we did a set of experiments, it's termed electrophysiology, where we're able to record the activity of a single neuron and look at a predefined pathway going to that neuron. Now, this pathway is called the prefrontal cortex to the amygdala. And one thing to note is that, let's just see if I can get this right here, is that there is also, just complicating it slightly, is there is this interneuron here as well. And so this is actually a GABAergic interneuron in the amygdala. Don't worry about it too much. It gets complicated rather quickly. And what we found is that the prenatal poly-IC was able to strengthen the prefrontal cortex input to the amygdala, whereas the postnatal LPS weakened that GABAergic interneuron. And when you do both of these, you see a combined effect, strengthening the PFC input and weakening that GABAergic interneuron. Now, the main takeaway from this is that we can see these effects of prenatal and postnatal immune activation that happened, a long time ago at the level of individual neurons and an individual neuronal circuit. So it leaves a kind of a long-lasting consequence to these particular neuronal circuits. Let's see. Apologize. All right. So the next, major question, and I think it's still a remaining question for us, is whether this phenotype is due to ongoing or developmentally sensitive immune dysregulation. So whether or not it was that initial immune insult that happens, prenatally or shortly after birth, and that's all that matters, or whether there are ongoing inflammatory processes as the animal becomes an adult. So the first evidence that there could be ongoing immune activation comes from looking at that resident immune cell, the microglia. And what we saw is by staining for microglia is that we could see an increase in the total number of these cells. So these cells are in green there. This is a technique known as immunohistochemistry that we're able to look at particular cells. And what we saw is overall an increase in this immune cell in the brain. And in a collaboration with Ori Kim, we looked at a number of inflammatory markers in the brain. Now, what we looked at is a number of different brain regions, the prefrontal cortex, amygdala, hippocampus, thalamus. And again, don't worry about these in particular. And we looked at a number of different inflammatory markers and some kind of more anti or immune regulatory markers as well. And then on the top ones, you can see this panel is for males and this bottom is for females. Now, overall, what we saw was an increase in a number of these pro-inflammatory markers within the brain. And this was true for both males and females. And we saw a decrease in some of the more immune regulatory or anti-inflammatory molecules in the brain in males, but actually an increase in females. So this is some pretty good evidence that there is ongoing inflammatory processes well into adulthood in these mice. So for the last part of this talk, I wanted to talk about just another line of research that really had gone back to when we were failing to see that model replicate. We kind of went back to the drawing board and we asked the question, well, what kind of assumptions is this model making? Because anytime you make a model, you make a set of assumptions. And one of the assumptions we were making is that poly-IC or that LPS was really perfectly mimicking that inflammatory risk. And we know that was unlikely to be true because we know that, in fact, there are really a wide variety of receptors in the body that are important for recognizing and responding to those invading microbes and inducing those inflammatory responses. And in fact, one of the prominent set of receptors are called the toll-like receptors. And in fact, poly-IC activates the toll-like receptor 3, LPS activates the toll-like receptor 4, but I was interested in another one of these toll-like receptors, toll-like receptor 7 or TLR7. And now this normally recognizes single-stranded RNA that's found in many types of viruses. But the reason I was particularly interested in TLR7 is if you go back to those risk factors, you'll remember it's not only bacterial and viral infections, but also things like autoimmune disorders can increase this risk as well. And the simplest way to tell you that TLR7 is involved in autoimmunity is to give you an example that if you give a drug that activates TLR7 every other day for 4 weeks, those mice will develop an autoimmune condition that looks a lot like lupus, characterized by things like the presence of auto-DNA autoantibodies and medical things like glomerulonephritis. Now, additionally, there's a lot of interest in TLR7 for its role in sex differences in the immune system, and it's encoded on the X chromosome. And it actually, so there's this process by which, with the X chromosome, you would essentially have 2 copies unless one is inactivated in females because there are 2 X chromosomes. But some genes will actually escape this, and its term, and will escape this X inactivation. And TLR7 just happens to be one of these genes, leading to sex differences between males and females. So what I designed was a fairly straightforward model, whereas instead of giving poly-IC or LPS, I gave something to activate TLR7 called imiquimod. And knowing that this drug actually activates or causes a much smaller immune response compared to poly-IC or LPS, I did repeated injections on embryonic day 12, 14, and 16 of pregnancy, and then assessed a battery of behavioral tests on the offspring. So first, I just want to show you that I was able to induce an inflammatory response with imiquimod. So this is just that pregnant mother. So this is just blood showing elevated cytokine. And interestingly, when I looked at the number of pups born, I actually saw a selective loss of female embryos. Now, this is an important caveat in this research, is that we have this skewed, these skewed litters and the skewed ones that survive. And so I'm not entirely sure what is leading to this selective loss of female embryos, but it's an intriguing finding nonetheless. Now, this time I assessed them on a really kind of comprehensive battery of behavioral tests. And I think when I went into this, I was, maybe expecting to find a more robust model, maybe get a more profound phenotype to work with. But what I found was something entirely different. So if you remember those pup USVs, now this time I actually saw a decrease instead of that increase where I found. I saw a significant decrease in both males and females. And when I looked at the USVs later on in life at 9 weeks of age, and this time I actually saw an increase with that social assent test, an increase in both investigation and the number of USVs emitted. When I looked at measures of anxiety-like behavior, I actually saw less anxiety-like behavior selectively in males, so there's more center time in the open field. And a similar trend on an Elevate Plus Mage, which is just another measure of anxiety-like behavior. Now, when I looked at social behavior, I found a really interesting effect. And I broke it down into the number of interactions, so the frequent, get my pointer, the frequency of interactions, the duration, and the duration per interaction. And what I found is that the mice that were treated with that prenatal imiquimod actually had more total number of interactions, but for a decrease in total time. Because what they were doing is they were doing these kind of brief spurts, where they're doing these hyper but fragmented social interactions. Now, at 8 weeks of age, I didn't see any effects in the females. But when I did a second test, a social dyad test, where I allowed the 2 mice to kind of freely interact, I saw a remarkably similar pattern in both males and females, where they were also exhibiting this kind of hyper but fragmented social behavior. So in summary, this time I saw a very different response with a decrease in the pup USVs, that kind of hyper but fragmented social behavior, and a decrease in anxiety-like behavior. And the last thing I wanted to tell you about in the behaviors is that when I looked at their activity patterns, and I looked at just how much they just move around at baseline, and that's often done in the open field, I didn't see a difference. But if you presented them with a social partner in the social interaction test, now they become much more hyperactive and moving around. And I'm not going to go into it today, but I did a whole circadian activity study and found that, relatively normal activity at baseline, but increased activity in the presence of circadian cues. And for the female mice, I found that you could see actually a much greater response to circulating hormones as well. So there's this form of kind of conditional hyperactivity, or in response to various stimuli. Now, the last thing we looked at is we looked at those resident immune cells in the brain, the microglia. We did IBA1 staining, and this time we stained for a lot of different brain regions. And overall, what we found was actually this time a decrease in the density of these microglia, really in a number of different brain regions, and really kind of across the board. And when we looked at these microglia in just a little bit more detail and did what's called a Scholl analysis, just looking at the change in morphology, or the change in shape of these immune cells, we saw that they actually became more branched. So this is something that can indicate the response to an immune challenge. And this is actually a little bit more signature of kind of a resting state. So overall, in this section, what we found was that kind of conditional hyperactivity. And now this time we actually saw a decrease in microglia density. Now, when I try to kind of put both of these models kind of together and look at these in the broader context, you'll see that we had this two different types of maternal inflammation, but they resulted in very different responses. Whereas that kind of prenatal TLR7 activation or that imiquimod produced kind of that hyper but fragmented, that kind of a little bit more kind of a frenetic kind of like tone, decrease in anxiety-like behavior. Maybe you can interpret it as more of kind of a risk-taking behavior. The poly-IC LPS resulted in a very different phenotype, with an increase in anxiety-like behavior, the decrease in social behavior. So you can see it's a little bit more of a depressive, less active phenotype. Now, what I think this really fits into the picture is, again, it's part of this accumulating evidence for this field, where there's increasing role for a potential immune involvement in both autism and many other psychiatric conditions. And what this particular research, I think, suggests is that all maternal immune activation may not be equal. And that, not only can maternal inflammation alter that trajectory of the developing nervous system, it might be able to alter it in very different ways. And I think ultimately what this might provide is really just kind of one clue as to how you can have this one risk factor of inflammation really be associated with such a wide array of psychiatric conditions. So thank you for your attention. I'd just like to quickly thank people who made this work possible. My laboratory at McLean Hospital. I worked under the laboratory of Bill Carlazon with a number of wonderful research assistants. This is a collaboration with Chris McDougall at the Lurie Center. And I would also just like to thank and send my gratitude again to the Brain and Behavior Foundation for their support and for the invitation to speak to everyone today.
Thank you very much, Galen, for the work you're doing, for your presentation today. Very, very interesting research. One of the questions that a few people ask and that I have is, is there work being done to look at interventions that might decrease the risks of this type of immune involvement or inflammation? What types of steps could be taken to decrease the risk that occurs?
That's a great question. And, and going along with that, there's two important things you need to think about. So if it is just that there is that inflammation early on, and that's the only thing that matters, then you'd need to intervene early. And there's been research looking at what are the particular immune molecules that can mediate some of this. And so there has been research suggesting that particular cytokines like IL-6 or IL-17 can produce some of these responses. But going along with that is, if there is ongoing immune dysregulation, can we then intervene later in life? And that's still an open area of investigation. And that's something that we had actually started getting into. And I know a couple of other laboratories have started to look at that. And I would say the answer there is still an open question. I don't think there's great evidence either way. I think there's some results suggesting that you can, and some that it doesn't have an effect.
Okay, and let me ask, with us all living through COVID right now, and especially for women who were pregnant during COVID, how does this fit in with concerns about somebody having COVID while pregnant?
Yes, I'm sure that's on a lot of people's minds. It turns out there was actually just a study that came out last week in a journal called, I think it was JAMA Network Open, if I'm remembering correctly, where they had looked at whether those that had been exposed to COVID or had a positive COVID pregnancy, or positive COVID test during pregnancy, and looked at the outcomes. And the problem is right now, of course, is it's still too early to tell. But there was in that study an increased risk, about a twofold increased risk for, a measure of neurodevelopment abnormality. But I will caution you in that, it is still too early for us to know. But the other thing to know, and fortunately, as far as I know, is that COVID and the coronavirus doesn't cross the placenta. So it would be more similar to some of the mechanisms that we see with the risk for influenza, as opposed to other types of viruses that can directly cross the placenta and affect the developing fetus. So in some regards, that's fortunate.
Yes, absolutely. Let me ask you one final question. Where do you see this work going in the next 5 years, next 10 years? What do you see as sort of next steps as we learn more about this important issue?
Yeah, I think it's also complicated right now. There are just so many factors, and it's hard to get to find out exactly what are all the necessary components for this to be true and for you to kind of see an effect. So I think the short term is really trying to nail down the exact components to produce this kind of increased risk. But then after we understand that a bit better, where it's going to be another layer of complexity, is then we're going to have to integrate this with genetics and the genetic risk, which we know is a huge component to these psychiatric conditions. And so it's that kind of gene by environment interaction that's really going to be, I think, a long set, and where I think we're ultimately heading.
Well, Galen, I want to again say thank you for the work that you've done on this, for spending time with us today on this presentation. So thank you very, very much.
Thank you. It was my pleasure.
Good, thanks. I also want to thank everybody who joined us today. And I'd like to ask people to please consider making a donation to BBRF today. 100% of every dollar donated for research is invested in our research grants. We're able to do this because our operating expenses are covered by separate foundation grants. This means that when you donate $1 for research, that dollar goes directly to the scientist. To make a gift, please visit our website, bbrfoundation.org, or call us at 1-800-829-8289. This webinar has been recorded. If you've missed any portion of the presentation or would like to share it with family or friends, please visit the events and webinars page on our website. And please join us again next month on July 12th at 2 p.m. Eastern Time, when Dr. Dawn Veligan, Director of the Division of Community Recovery Research and Training and Chair of the Department of Psychiatry at the University of Texas Health Science Center, San Antonio, will present When Medicine is Not Enough for Schizophrenia and Psychosis. Thank you, and remember, together we can dramatically improve the lives of those living with mental illnesses and enable more people to live full, happy, and productive lives. Take care. Bye.